I want to introduce our second speaker, who will be Paul Modrich. He was born a little bit further away from here in a small town called Wayton in New Mexico in the United States in 1946. He got his PhD at Stanford University in 1973, and he's now an investigator at Howard Hughes Medical Institute, and he's also a James B. Duke professor at Duke University in Durham, North Carolina. And with this, I welcome Professor Modrich to the stage.
Thank you. So I want to thank the Royal Swedish Academy for their acknowledgement of DNA repair and for permitting me to share in this wonderful honor. So, as Dr. Lindsay said, we've studied mismatch repair, which corrects base pairing errors in the DNA helix and plays an important role in the control of mutation production. Inactivation of the human pathway increases the mutation rate 100 to 1,000-fold, and the resulting consequences can be pretty dramatic, and I'll cite several examples as I go along. Oops, this is not working. Oh, this one, sorry. So I thought I'd begin with a little history. The idea that mismatched base pairs occur in cells and that such lesions trigger their own processing was suggested 50 years ago by Robin Holliday in the context of genetic recombination. Breakage and joining of DNA helices was known to occur during this process, and Holliday pointed out that the precision of rejoining could be assured by formation of a heteroduplex joint, a region of helix in which the two strands are derived from the different recombining partners. He also pointed out that if this heteroduplex should span a genetic difference between the two DNAs, it will contain one or more mismatched base pairs. And he invoked processing of those base pairs as the basis of the recombination-associated phenomenon called gene conversion, noting that if there are enzymes which can repair points of damage in DNA, it would seem possible that the same enzymes could recognize the abnormality of base pairing and by exchange reactions rectify this. Direct evidence that mismatched base pairs trigger a repair reaction was provided by bacterial transformation experiments, and our interest in this effect was prompted by the E. coli studies done in Matt Melselson's lab. Wagner, and using artificially constructed heteroduplexes containing multiple mismatched base pairs, Wagner and Melselson showed that mismatches trigger a repair reaction when introduced into the E. coli cell. They also showed that closely spaced mismatches separated by 1,000 base pairs or so, are usually repaired on the same DNA strand. And based on that strand bias effect, they suggested that in addition to its role in genetic recombination, mismatch repair may act to correct mutations that arise as replication errors. If so, it may be that mismatch repair acts in a directed manner in conjunction with sistered chromatid exchange, or that it occurs with particularly high efficiency on newly synthesized DNA strands, possibly because of their undermethylation or because of a special relation to the replication complex. This proved to be a particularly insightful proposal. We now know from the work of many labs that correction of DNA biosynthetic errors is a primary job of mismatch repair. And in order to function in that manner, the repair system has to be able to do two things. It has to recognize the mismatch produced by the replication error, but it must also be able to identify the new strand which contains the mistake. And Pat Pukila, working in the Messelson lab, showed that strand direction of E. coli mismatch repair is dictated by the state of adenine methylation at GATC sequences, because this modification occurs after DNA synthesis, newly synthesized DNA exists in a transiently unmodified form, and it's this transient absence of methylation that directs mismatch repair to the new strand. And Barry Glickman, Miro Radman, and others showed that this methyl-directed reaction depends on the products of 4 mutator genes, MutH, MutL, MutS, and UVRD. And activation of any of these genes increases the mutation rate in the E. coli cell by 50 to 100-fold, indicating the importance of this pathway and mutation avoidance and genetic stability. So this is where we entered the picture. I was interested in how mismatches might be recognized, how the state of DNA methylation at one side on the helix could direct mismatch repair somewhere else. And to address those questions, we needed a biochemical assay. So exploiting several tricks developed in Norton Zinder's lab, Ali and Liu built heteroduplex DNAs like these, circular molecules in which the strands were in defined states of DNA methylation, and which contained a GT mismatch in the recognition site for EcoR1 restriction enzyme. Because this mismatch blocks cleavage by EcoR1, when this DNA is digested with EcoR1 and BAMH1, which cleaves down here, the product gives you a full-length linear product, which runs on an agarose gel here. But if repair occurs on the unmethylated strand as predicted by the Mendelssohn mechanism, then this GT mismatch will be converted to a GC base pair, restoring EcoR1 sensitivity, so digestion with the 2 enzymes will yield 2 smaller fragments that run down here. In fact, incubation of this DNA with extracts prepared from broken E. coli cells converts it to an EcoR1 sensitive form. And as predicted by the Mendelssohn mechanism, this reaction is blocked when both DNA strands are methylated, and the extract reaction also depends on the products of MutH, MutL, MutS, and UVRD, which I mentioned a moment ago are required for mismatch repair in the bacterial cell. Michael Su, Bob Lehue, and Karen O showed that this extract reaction supports the repair of all the base-base mismatches except CC, and that at least one hemimethylated GATC site is required for repair to occur. And that latter observation prompted simplification of our substrates to molecules that contain a mismatch in a single GATC site that are separated by about 1,000 base pairs. And Ali and Liu showed that this extract reaction is accompanied by DNA synthesis occurring on the unmethylated strand, which suggested that mismatch repair was occurring by an excision repair mechanism. And working in collaboration with Jack Griffith's lab at the University of North Carolina, Mike Su and Michel Grilli confirmed this to be the case. So when hemimethylated DNA is incubated with an E. coli extract under conditions of a DNA synthesis block, this results in production of a single strand gap that spans the shorter path between those 2 DNA sites. And this was true for both hemimethylated configurations of the substrate. Because the 2 DNA strands are anti-parallel, this said that there's no obligate polarity of the 2 DNA strands, and suggested that the methyl-directed system is capable of bidirectional excision. And I'll return to that point in a moment. Well, just to try and figure out how this reaction works, we isolated the MutH, MutL, MutS, and UVRD gene products. We knew from the work of Peter Emerson's lab that UVRD encodes an enzyme called DNA helicase 2 that unwinds the helix in an ATP-dependent fashion, but the nature of the other 3 proteins was not known. And Michael Su quickly showed that MutS is responsible for mismatch recognition, a conclusion that's beautifully confirmed in this crystal structure of a MutS dimer bound to a GT mismatch that was solved in Tisha Sixman's lab. Michel Grilli showed that MutL is recruited to the MutS mismatch complex in an ATP-dependent fashion, but otherwise essentially does not alter the covalent nature of the DNA helix. And Kate Welsh and Ali and Liu found that the MutH gene product has a very tightly associated but nearly dead endonuclease activity that incises DNA at an unmethylated GATC sequence. And I'll tell you in a moment that latent endonuclease is activated by assembly of this complex. But these 4 proteins were not sufficient, so using biochemical and genetic approaches, Karen O and Bob LeHue used genetic and biochemical approaches to identify other required activities. And they identified 4. An enzyme called exonuclease 1, which hydrolyzes single-stranded DNA with 3' to 5' polarity. The single-strand DNA binding protein, SSB. DNA polymerase III holoenzyme, which functions in chromosome replication, and DNA ligase. Supplementation of MutH, MutL, MutS, and UVRD, or DNA helicase 2, with those 4 proteins was sufficient to reconstitute methyl-directed repair in a purified system. And I'll tell you in a second why sufficient here is in quotes. The assay shown here is a refined version of the one I showed earlier. In this case, the GT mismatch is placed in overlapping recognition sites for 2 restriction enzymes. It blocks cleavage by both enzymes, but when repair occurs on the bottom strand, the GT mismatch is converted into an AT base pair, yielding a good Hindi 3 site, and when repair occurs on the top strand, the mismatch is converted to a GC base pair and a good XHO1 site, and this permits repair to be directly scored on either strand. Anyway, using that kind of assay, heteroduplex DNA that lacks a GATC site is not a substrate for repair in this system, nor is a heteroduplex in which both strands are methylated. However, hemimethylated DNA is repaired, and when the methyl group resides on the strand containing the mismatch G, repair is exclusively to GC, and when the strand containing the mismatch T is methylated, repair is to AT. In other words, repair is methyl-directed. Now, as I mentioned a moment ago, analysis of the extract reaction suggested that the methyl-directed repair might have a bidirectional excision capability, which would presumably depend on both 5' to 3' and 3' to 5' exonuclease activities, and that proved to be correct. Deanie Cooper, Vickers Burdett, and Celia Batinger showed that when the unmethylated strand the unmethylated GATC site resides 5' to the mismatch, repair depends on a 5' to 3' exonuclease, or depends on 5' to 3' hydrolysis that can be mediated by one of 2 exonucleases, XO7 or RECJ. And collaborative experiments with Sue Lovett's lab showed that when the GATC site is located 3' to the mismatch, repair depends on 3 to 5 excision by exonuclease 1 or exonuclease 10. Our original reconstituted system shown here contained only 1 exonuclease, exonuclease 1, and the reason these experiments were successful was because the DNA polymerase III holoenzyme preparations used in these experiments were contaminated by XO7. So let me tell you how this reaction works. Mismatch recognition by MutS leads to recruitment of MutL, as I mentioned. And MutL serves to interface mismatch recognition by MutS to activation of several downstream repair activities. And one of those is the latent MutH GATC endonuclease I mentioned a moment ago. Karen O showed that methyl-directed repair initiates by activation of MutH in a mismatch MutS-MutL dependent fashion. Incision by activated MutH is directed to the unmethylated strand and can occur either 5' or 3' to the mismatch, consistent with a bidirectional mechanism. But the important point, and the one I'll ask you to keep in mind because I'll come back to it in a moment, is that it's the resulting MutH strand break that is the real signal that directs repair in this system. MutS and MutL also activates the excision system, which is comprised of DNA helicase 2, the UVRD gene product, and the several single-strand exonucleases I mentioned a moment ago. Vivian Dow and Miyuki Yamaguchi showed that MutS and MutL load helicase 2 at the mutate strand break, so unwinding proceeds back towards the mismatch. And this is true for both heteroduplex orientation, whether the strand break is located 5' or 3' to the mismatch. The single-strand DNA segment that's displaced by helicase unwinding is then degraded by one of these single-strand exonucleases I mentioned, XO7 or RECJ, when the break is 5' to the mismatch, or by XO1 or XO10, when the break is 3' to the mismatch. And the resulting gap is then repaired by DNA polymerase III holoenzyme, and although not shown here, ligase finishes its job by restoring covalent continuity to the helix. So, and I just want to mention one thing. So, and Anna Plushinic showed that these function of these components of the polymerase III holoenzyme absolutely restricted to the repair synthesis step. They have no function in the mutate activation or the excision steps of the reaction, and I mentioned that in passing because the human reaction I'm going to talk about shortly differs in that regard. So this system clearly involves action at a distance, coupled interaction of two DNA sites that in our substrates are separated by about 1,000 base pairs. But the mechanism by which that occurs has been a subject of debate for more than 20 years. This orientation-dependent loading of helicase shown here implies that the repair system can establish the relative orientation of those two sites. And that requires that a signal be transferred between those two sites along the helix contour. Several labs, including ours, have shown that mut-S and probably the mut-LS complex is capable of moving along the helix in an ATP-dependent manner. And we favor the idea that that sort of movement is involved in this signaling, but that's not proven, and because my time is limited, I'm going to leave it at that. So as our work on the methyl-directed reaction progressed, we became curious whether a similar strand-directed reaction might occur in higher cells. Work from a number of labs had shown that mismatches are rectified in higher cells, but there was no evidence for strand-directed repair. And the problem we had addressing this question was we had no idea what the strand signals were. Jude Holmes, who began these experiments, circumvented that problem by exploiting an observation we'd made in the E. coli pathway, namely that the purpose of the hemimethylated GATC site and mut-H is simply to provide a strand break. The strand break is the real signal. So Jude built heteroduplex DNAs containing a site-specific, strand-specific nick, and he showed that these DNAs are subject to repair on the nick strand and extracts of broken human cells. Very little repair occurs on the continuous strand, and when both strands are covalently continuous, repair is blocked. And Wei Hong Fong showed that the DNA hydrolytic events occurring on these molecules is restricted to the shorter path between the two DNA sites, regardless of their 5' to 3' orientation. And this is reminiscent of the bidirectional nature of the E. coli reaction I mentioned a moment ago. But as you will see, the mechanism of the human reaction differs significantly. So the development that permitted us to identify the key proteins involved in initiation of human mismatch repair and pursue the mechanism of the reaction was the publication of two papers from the De La Chapelle, Vogelstein, and Peruccio laboratories. And these studies showed that frequent mutations in simple DNA repetitive sequences are characteristic of tumors that appear in patients with Lynch syndrome, a common hereditary cancer that accounts for about 5% of colon cancers in the clinics, as well as about 15% of sporadic cancers with Lynch-like features. And we knew from previous work of Levinson and Gutman that these kinds of mutations are also characteristic of E. coli mismatch repair mutants, so it seemed plausible to us that these tumor cells might be defective in mismatch repair. So we acquired a number. One thing I forgot to mention is, what did I do? Yeah. I forgot to mention this. This phenotype, that is frequent mutations in these simple repetitive sequences called microsatellite instability. So we acquired a number of tumor cell lines, with microsatellite instability, initially from Bert Vogelstein and then from a number of other labs. And Goman Lee, Wei-Hong Fong, Matt Longley, and Jim Drummond showed all of these cell lines to be defective in the strand-directed reaction I mentioned a moment ago. And availability of these repair-defective cells permitted us to isolate and identify the proteins that are deficient in these lines. Jim Drummond and Goman Lee showed that several of these lines are deficient in a protein we call mut-S alpha, a heterodimer of two mut-S homologs, MSH2 and MSH6, that we now know to be the primary mismatch recognition activity in human cells. And Goman Lee showed that the remaining lines were defective in a protein we call mut-L alpha, a heterodimer of two mut-L homologs, MLH1 and PMS2. While we were pursuing these biochemical experiments, several labs, primarily those of Bert Vogelstein and Richard Klaudner, were sequencing mut-S and mut-L homolog genes in Lynch syndrome families, and they showed that mutations in these genes segregate with the disease phenotype. And it's now very clear that the vast majority of cancers in Lynch families are caused by inactivation of one of these two heterodimers. The last four cell lines shown here proved to be exceptional. Each was derived from a microsatellite unstable sporadic cancer. Each is defective in mismatch repair. And Jim Drummond showed that all four of these cell lines failed to produce the MLH1 subunit of mut-L alpha, and that repair could be restored to extracts in each of those cases by addition of purified mut-L alpha. But we encountered some skepticism when we shared these findings with our sequencing colleagues, who told us that they'd sequenced the MLH1 gene in this line and this line, and it was normal. But Sandy Markowitz, with whom we were collaborating on the two vagal lines shown here, took us seriously, and collaborative experiments largely done in his lab showed that the MLH1 gene in these four cell lines is epigenetically silenced by CpG methylation within the promoter. This blocks transcription. The MLH1 polypeptide is not produced, resulting in a mismatch repair defect. And the proof of that is shown here. Exposure of these cell lines to 5-azacitidine, which results in transient cytosine demethylation, results in transient MLH1 expression. It goes up and then it comes back down. And I mention this effect because it's very important in the clinics, where it's believed to account for about 15% of colon cancers. So I want to devote the remaining minutes to telling you what we've learned about how human mismatch repair works. The 8 proteins shown here are sufficient to reconstitute a minimal system that supports bidirectional human mismatch repair in a purified system. And I want to tell you about 2 strand-directed reactions that we've identified that are supported by subsets of these proteins. The first of these shown here is essentially, in a sense, a trivial example of strand direction. The only exonuclease that has been definitively implicated in eukaryotic mismatch repair is an enzyme called exonuclease 1, which hydrolyzes duplex DNA with 5' to 3' polarity. Yolk and Shell showed that EXO1 is activated by mut-S alpha, the human mismatch recognition activity I mentioned a moment ago, in a mismatch-dependent manner. And we think that movement of mut-S alpha along the helix is involved in coupling action at these 2 sites. Activation by mut-S alpha renders EXO1 highly processive, and processive action of the mut-S alpha EXO1 complex is controlled by the RPA single binding protein, which displaces this complex from the helix after removal of about 200 nucleotides. This RPA-filled gap is an incredibly poor substrate for EXO1, and the enzyme cannot reload without the mismatch-dependent assistance of mut-S alpha. What this means is that this iterative, and I'm not sure I mentioned it, so excision by this processive complex when it's being controlled by RPA is restricted to about 200 nucleotides. And as I said, this gap, RPA-filled gap, is incredibly poor substrate for EXO1, and it requires the assistance of mutas alpha, mismatch-dependent assistance of mutas alpha to reload. So what this means is that this iterative cycle of removal of about 200 nucleotides will continue until the mismatch is gone, at which point excision is dramatically attenuated because mutas alpha can no longer assist in this way. This reaction does not depend on the other mismatch repair protein I mentioned, does not require the other mismatch repair protein I mentioned, mutal alpha, but mutal alpha does modestly enhance the mismatch dependence of this effect. The second strand-directed reaction that I want to tell you about is more interesting. Fried Kadyrov, Leo Dezantiev, and Nicoletta Constantine showed that unlike bacterial mutal, human mutal alpha is a latent endonuclease that's activated in a manner that depends on a mismatch, mutas alpha mismatch recognition activity, the PCNA replication clamp, and RFC, the clamp loader that places PCNA on the helix. An action of this endonuclease is strand-directed. It's targeted to the heteroduplex strand that contains the pre-existing break. Incision by mutal alpha can occur at multiple sites along the helix, but initial events are biased to the distal side of the mismatch. Fried showed that this reaction occurs on both 5' and 3' heteroduplex to yield products, molecules in which the mismatch is bracketed by 5' and 3' strand breaks. And he showed that 5' termini produced in this manner can serve as loading sites for two modes of mismatch removal: hydrolytic removal by mutas alpha activated EXO1, the reaction I mentioned a moment ago, or synthesis-driven strand displacement by DNA polymerase delta, and other potential excision mechanisms are possible as well. So we think that this endonuclease function of mutal alpha is a primary function of that protein and eukaryotic mismatch repair. And the reason I say that is shown here. Fried identified a metal-binding endonuclease active site motif within the PMS2 subunit of mutal alpha, this motif shown in red here. Amino acid substitutions within this motif have no effect on assembly of the mutal alpha mutas alpha DNA ternary complex, but they inactivate endonuclease function and they abolish mismatch repair in human, yeast, and mouse cells. And in the case of mice, this is associated with strong cancer predisposition. I should add that the yeast experiments shown here were done in collaboration with Tom Kunkel's lab, and the mouse experiments with Winfried Edelman's lab at Albert Einstein. So this red motif, this endonuclease motif, is conserved in eukaryotic PMS2 homologs, but it's also found in many bacterial mutels. The notable exceptions are mutal proteins from bacteria like E. coli and its cousins that rely on GATC methylation to direct mismatch repair. And in those bacteria, this motif is conspicuously absent. So this is my last experimental slide. So I want to say just a little bit about what we've learned about activation, the mechanism of activation of mutal alpha. Anna Prushinik and Leo Dezantiev showed that the only function of the pre-existing strand break is to provide a loading site for the PCNA clamp, and that RFC involvement in this reaction is restricted to loading PCNA on the helix. So PCNA, mutas alpha, and mutal alpha are the players here. And loaded PCNA does two important things. Physical interaction of PCNA with mutal alpha is required to activate endonuclease function, and the strand direction with which PCNA is loaded on the helix determines the strand direction of endonuclease action. And that's depicted here in a schematic form. The two faces of the PCNA clamp are not equivalent. The O'Donnell and Korean labs have shown that PCNA is loaded at 3' double-strand, single-strand junctions with a unique orientation. That's depicted here, where the gold face is oriented towards a 3' end. We know from the work of Joe Jerichny and Richard Kolodner that mutas alpha is tethered to the replication fork via physical interaction with PCNA. And we know that both of these proteins can move along the helix. When mutas alpha recognizes a mismatch, this leads to recruitment, and I think I pointed there, leads to recruitment of mutal alpha, which is capable of interaction with both PCNA and mutas alpha. And the idea is that the asymmetry of PCNA loading is preserved in the PCNA mutal alpha complex, and this serves to orient the endonuclease active site relative to the two strands of the helix. So when I began this discussion of eukaryotic mismatch repair, I mentioned that we had no idea about the nature of the strand signal, but those two strand-directed reactions that I've described suggest several possibilities in the context of the eukaryotic replication fork, which is shown here. For example, 5' termini on the lagging strand would presumably support loading of mutas alpha activated XO1, and genetic studies from Tom Kunkel's lab is consistent with that view. I also mentioned that strand direction of mutal alpha endonuclease action is determined by the orientation with which the PCNA clamp is loaded on the helix. And at the fork, that is determined by 3' termini on the leading and lagging strand. So an obvious possibility is that termini that naturally occur at the eukaryotic replication fork may well serve as default strand signals that direct mismatch repair in the cell. So that's all I have to say. I'm going to stop there. I've acknowledged the people that did the work I've described. Some of them returned to Duke for a recent party. And this is a complete list of my colleagues and collaborators in this work. One of these collaborators is also a friend, Aziz Sanchar, our next speaker. Thank you.